Okay, so now we have a problem. I don't know whether you see it or not. The problem is our dendritic cells basically activate our adaptive immune system. They activate it via MHC2 molecules. So do our dendritic cells have MHC1 molecule? And if they do have MHC1 molecule, it will make them a target of cytotoxic T cells. If that happens, you can very well imagine if our dendritic cells are killed by cytotoxic T cells on the onset of the infection, it will basically kill the immune response because our dendritic cells are the ones which are activating basically the adaptive immune system. So what is the trick here? We are going to talk about that. But first of all, let me tell you how T cells are selected. We are going to select T cells, those T cells which are going to be useful, the ones with TCR that does not recognize self molecules, but however it can interact with MHC molecules. So let's see how that happens. So basically we don't want to keep T cells or useless cells that are not going to do any useful work. So any T cell that does not express TCR is killed by default. Okay, so those T cells which fail to produce TCR are killed, so we don't have to keep them around, feed them and take care of them when they're completely useless. TCRs with no recognition of self MHC or self peptide, they also go through apoptosis because these T cells, although they have a T cell receptor, but their T cell receptor does not recognize self MHC or self peptides. We don't need these type of T cells either. T cells which can bind self MHC molecule and self peptide, this interaction is strong. This type of T cell can cause an autoimmune reaction, meaning if recognizes strong interaction, basically recognition means that T cell is going to become activated. You don't want any T cells that can recognize self molecules by strong recognition and destroy them. So these T cells will also be eliminated. So we are eliminating T cells that are useless to us or can cause damage to the host tissue. T cells, however, with weak recognition of self MHC and self peptides, weak recognition, these T cells are going to be allowed to live. And these T cells, since with the weak interaction, these T cells will not be activated. These T cells are useful. They will be allowed to live. Their survival, maturation will also occur in due course. This is basically the positive signal. This positive signal is for the survival of these type of T cells that have weak recognition of self MHC and self peptide molecules. The one here is negative selection. It basically is getting rid of T cells that are not going to be useful to us. So now the question is, how is it that our dendritic cells do not get killed by cytotoxic T cells? First of all, let's see how our other body cells, how they are destroyed, how they are able to display antigens or small peptide fragments on their surface in association with MHC1 molecule. That will clarify some of the things, I hope. So a normal cell, a body cell that gets infected by a particular virus, the virus, when it infects the cell, when it enters the cell, it will be released, or its components will be released in the cytoplasm, because this virus needs machinery, cellular machinery, to replicate itself. So when the virus is released in the cytoplasm, our cellular machinery will make viral proteins, which are shown in red here, and viral RNA will be used to make these proteins. It so happens even in our normal proteins, most of the proteins, about 30% of the proteins, they are degraded by our cells. This is a normal process. How they are degraded, we talked about this some time back also. For example, proteins that have to be degraded are attached to ubiquitin. Any protein that has a ubiquitin molecule attached to it is guided towards proteasome. This is basically a small complex of molecules which basically degrades proteins into small fragments, and these fragments are about the same size that can fit in the groove of MHC molecules. So this viral protein will also get degraded. It will get degraded and it will be pumped into ER, endoplasmic reticulum, through a transporter called ABC transporter, which is shown in red right here. These peptide fragments, whether they are viral or normal cell peptides, when they enter this ER, they will find the MHC class 1 alpha chain, and it will bind, these fragments will bind this MHC1 molecule. This binding is required by MHC molecule to survive. If this binding doesn't take place, MHC1 molecule, class 1 molecules will be degraded. So these MHC1 molecules will ultimately be passed on to Golgi apparatus. We have seen that happens. Golgi apparatus will process these molecules, and ultimately they'll be put in a vesicle destined for the plasma membrane. So this is basically the route which is taken by the proteins, peptide fragments, normal peptide fragments of our cells, or cells that have been infected by viral proteins. Our cells have flags on their surface. These flags could be white flags showing the proteins that the cell carries, only normal proteins that are present in the cell, or these flags can be red. These red flags basically are MHC1 molecules that are being displayed on the surface of the cell, and our cytotoxic T cells can read, they're running around reading the surface of the cells. Wherever they see a red flag on the surface of the cell, they will destroy it. The cell will be destroyed. We have already seen two different mechanisms how T cells are able to destroy a target cell. Also, I would like to mention that the main point here is the proteins are made in the cytoplasm and they're imported into the ER. When we compare it with the other system, how MHC2 molecules acquire peptide fragments, it will be a different strategy than this. So the antigen presenting cells, remember, are phagocytic cells. They engulf particles or the pathogens and they put them into a phagosome. This phagosome also, or early endosome, this early endosome matures into late endosome, and the contents of this vesicle never leave the vesicle in which they are contained. For example, this red ball and green ball joined together, you will never see them come out into the cytoplasm. They will stay in endosome. Here in the Golgi apparatus, remember, our MHC2 molecule is a protein that goes to the surface. All proteins that go to the surface are processed through ER and Golgi. So Golgi apparatus will make a vesicle, and this vesicle will fuse with the early endosome, forming the late endosome. Now the peptide fragment, which is a foreign or pathogen in origin, is in the phagosome is in the late endosome along with the MHC2 molecule. Initially, the MHC class 2 molecules, when they're processed in Golgi apparatus or the endoplasmic reticulum, their site is blocked by an invariant chain shown here in blue. This is the invariant chain. It is binding the location of MHC2 molecule where peptides bind. So when this fusion takes place of the vesicle originating carrying the MHC2 molecule and the early endosome, late endosome is formed. Here the rest of as we know that they will also fuse with the lysosome, and proteins are degraded there, except for a small little fragment of the invariant chain which is still bound to the MHC2 molecule. This small invariant chain fragment will be removed by a protein called HLA-DM. Now our MHC2 molecule has the ability to bind a protein, and in this case it will bind the protein which was brought in by the phagosome, this little red dot right here, and this MHC2 molecule will now be sent to the cell surface, displaying this foreign peptide, and which will be used to activate helper T cells. I hope you remember that. So now there is a difference, that the difference is here that these viral proteins are never in the cytoplasm to be able, so these proteins cannot bind the MHC class 1 molecule. This is why our antigen presenting cells will not be destroyed by cytotoxic T cells. So our immune system does not destroy the antigen presenting cells, the cells which initiate immune response and are trying to help our body initiate an immune response. So these are two different strategies cells have taken to display protein fragments via MHC1 and MHC2 molecules.